Hello everyone, let's study the somatic embryogenesis week. Somatic embryogenesis it occurs during plant tissue culturing. It is the process of embryo initiation and their development from vegetative or we can say non-gametic cell. So there are two types of cells present in the mature pollen grain, which are those vegetative cell and generative cells. So the vegetative cell is bigger in size and it is the food reservoir, but the generative cells, which the de novo synthesis of zygote is by the fusion of generative cell with the female gametophyte, and then there is formation of zygote. But in the somatic embryogenesis, what is happening is this vegetative cell, from these vegetative cells we are forming the embryos. So as a process where embryo is derived from single cell somatic cells, so this vegetative cell is haploid, we can say. Sorry, we cannot say haploid, but it is the single cell. From the single cell we are forming the embryo. Somatic embryos are formed from plant cells that are not normally involved in embryo formation. As I mentioned, this cell is only the food reservoir. It is not involved in the formation of embryo, but it is involved in the formation of endosperm. So from this we are developing the embryo. That's why it is somatic embryogenesis. And in zygotic embryogenesis, it is the formation when the female gamete fuses with the male gamete. Then there is formation of zygote, which give rise to embryo. Then the types of somatic embryogenesis. There are the two main types. First one is direct somatic embryogenesis. What we need to keep in mind that here there is no formation of callus, so that's why it is the direct somatoembryogenesis. The explants are capable due to they are having these pre-embryonic determined cells, and which give rise to the embryo. As they are committed to the formation of embryo, that's why we are saying it is the direct embryogenesis. These cells are present in scutellum of cereals, ovules and zygote, etc. As all these are the embryogenic tissues. In the indirect somatic embryogenesis, there is a formation of callus. Now, from the callus formation, further stages are progressed or developed. That's why it is indirect somatic embryogenesis. When the explant is produced from the callus, which is maintained or differentiated into the embryo, and the cells in this callus are having the induced embryogenetic determined cells. As the specific growth regulators and culture mediums we provide to callus, there is redetermination of embryogenic development, and this redetermination is in the induced embryogenic determined cells. So those cells are called as IEDCs. Let's study this diagram for a clear concept. So here is the direct. We take the explant, we extract the cells, somatic cells, and we give rise to the somatic embryo, and it further grows into seedling. In indirect, we take the immature embryo, and then there is the formation of callus, and from the callus, due to these induced embryogenic determined cells, the somatic embryo is formed, and then there is formation of root and shoot. But in de novo, what is occurring, how the embryogenesis is different from organogenesis, as we are taking a single cell here, but we are taking the tissue, or we can say meristematic cells. We are taking the cells. So that's how it is different from organogenesis. Let's see the stages of embryogenesis. The first stage is the induction. So for the induction, auxin plays an important role, particularly this 2,4-D, which induces the embryogenesis. And the requirement of these exogenous auxins depends on the nature of explant we are using and their relative concentration to the auxin. Development is the second stage. First stage was induction with the help of auxin. Second is development. Here after the reinitiation of cell division, the period of cell proliferation in the presence of auxin, the auxin embryogenic cells are released into the auxin free medium. So now for the development we are providing the auxin free medium. And for the maturation, as we are not growing them or culturing them in the natural environment, but we are providing them the natural environment, which is nothing but artificial, we can say. But so that's why there is the loss, or we can say very poor germinability, which is nothing but the conversion into plant, so that their maturation is kind of low. Their maturation ability is low. So that's why we are providing the stress hormone here, which induces or which triggers the expression of genes which are which promotes the embryogenesis, or which carry out the normal development of the embryo. So here we can see the all stages and steps. So from the callus, here this is the callus formation. From the callus, this is I think indirect step. Then the formation of embryogenesis. Here this one is the heart shape, torpedo shape, and then plantlets. So that's how we produced a plant from single cell. The stages: globular, heart, torpedo, and cotyledon. The factors affecting are polyamides, as as we are producing in vitro, in vivo somatic embryogenesis. Polyamides are required for cell growth, cell proliferation, and their aging. So three main polyamides here are important: putrescine, spermidine, and spermine. This putrescine shows the most drastic increase in somatic embryogenesis. So we use this more. Genotypic effects, how these affect, as there must be some there must be some endogenous levels of hormones which are getting affected, so they may cause some genetic variations. The genotypic effect on somatic embryogenesis occur as for regeneration via shoot or bud differentiation. Hormones level and other factors causes the genetic variation. Synthetic seeds, this is the main application why we are doing the formation, or why we are going for the somatic embryogenesis. This is the main reason to get the synthetic seeds. So how we get it? By the two types. First is desiccated and second is hydrated one. In desiccated one, what we do is these seeds are produced naked or in the polyoxyethylene glycol encapsulated somatic embryos. So somatic embryo is encapsulated in polyoxyethylene glycol and it is produced in desiccation tolerant species. Hydrated synthetic ones are encapsulated in hydrogel. So somatic embryos are encapsulated in hydrogels like sodium alginate, potassium alginate, and gelatin, sodium pectate, etc. So this is the whole process of somatic embryogenesis, and these are the desiccated hydrogel. What was that? Hydrated synthetic seeds. They are in the somatic embryos are in the hydrogel sodium alginate. Here it is sodium alginate. And these factors we study. Other factors are auxins and cytokinins. Cytokinins, what they do, they inhibit the embryogenic potential. Auxin is inducing and cytokinin is inhibiting. Next is nitrogen. If we use the nitrogen in the form of NH4Cl with the KNO3, it will induce the embryo development. And polyamides we studied, and that's all. So all this was brief intro of somatic embryogenesis.